Anyway, thanks everybody for coming today, and thanks for having me. Thanks to everybody that's made this event possible and all the wonderful food, and especially David there in the back. As Tree said, my daughter was diagnosed with diabetes about 12 years ago. As a parent, you get really upset. As a researcher, you try to understand why. And at the time, when she was born in 99, at the time, microbiology wasn't anywhere it is today. And if you pick up a newspaper or magazine or read Eating Well magazine, things that Gretel writes, you have to live under a rock not to know that the human microbiome or bacteria control a significant part of our health and well-being. And that's mainly because of changes in technology. The cost of sequencing has gotten cheaper, and the scientists have gotten a lot smarter, and granting agencies have started putting money into bugs. So now they've been connected. You name an ailment, there's a bug probably connected to it in some form or fashion. So I started trying to understand from an evolutionary perspective where humans and microbes kind of evolved in our symbiosis over millions of years. And so I slowly started to kind of dig my way into the discipline, and I started going to conferences, started presenting papers, started publishing papers, and kind of came in it kind of backwards. And so I started something called the Human Food Project, which is basically a blog and a Facebook page and a lot of hours and a lot of hard work. And it's an attempt to try to blur the line between science, metagenomics, and the microbiome and people's understanding of it. So typically there's a 10 to 20 year lag between what science says and what the public understands about that science and making it operational in our lives, and specifically in regard to public policy. So I wanted to try to create basically a vehicle to fast track that information to individuals like you, especially young parents who don't know what they're doing when they're having children, just like when we didn't know what we were doing when we were having our children. So today I'm going to talk about, and again, it's kind of a loose format, so if anybody has a question actually while I'm talking, just raise your hand. And we'll just kind of make it interactive. If anybody's confused about something, which might happen once or twice during the talk, or if you have a question about something that I'm saying, just raise your hand and we can stop and talk about it, and we'll move on to the next slide. And let's see if we can get this thing to work. So today we're basically going to be talking about the gut. So this is the colon. The colon's the last 5 foot of your gastrointestinal tract, and the business end of that, the last foot, is what we're most interested in. So if anybody, again, I'm just going to gloss over a lot of things here. If anybody has any questions, just raise your hand. So most of us in this room know that we have bacteria in our body and on our body. The bacteria from our mouth to our anus weighs about 3 pounds for a healthy person. It weighs about the same as a human brain. If you remove the brain from the human body, it's probably not going to work so well. If you remove those 3 pounds of bacteria or a fraction of them or impact them in any significant way, the overall system's not going to work that well. And so it's now considered a metabolic organ, just like a liver or heart or pancreas. So we now know that impacting that can change our life in a dramatic way. So why should we care about our microbes? They determine whether Tylenol is toxic to your liver. Okay, so that's just one simple thing. The other one would be they tell you to have they tell you who to have sex with if you're a fly. So flies determine who they're going to have sex with based on the bacteria on that fly. You can shift the bacteria on that fly. The fly no longer has sex. They steal genes from your food to help you digest it. And the list goes on and on and on. And so the bacteria, again, most people in this room would know that the bacteria plays some role in digestion. You do not possess some enzymes to break down dietary fiber, for example. Just like a cow, you possess the bacteria deep in your gut that'll break those oligosaccharides and polysaccharides and those things down, turn it into short-chain fatty acids so you can use it. Again, if you remove those bacteria from your gastrointestinal tract, and about 99% of them live in your large bowel, the rest of them are scattered throughout the stomach and small intestine, and then your body's covered in them as well. You can put 1 billion bacteria on the tip of a pen, by the way, or a period at the end of a sentence is what 1 billion bacteria looks like if you stack them all on top of one another. And again, E. coli is a bacteria that we all hear about. It's in the news all the time. It's hurting people sometimes. It's a regular member of the gastrointestinal tract, but there's more E. coli in your gut than there are people on Earth. And again, if you look at people in general, you pick any two humans, it doesn't matter who you are, we're all basically 99.9% similar with regards to our genomes. However, you can take any 2 E. coli out of your gut, and they could be as much as 40% difference. So you're not a beautiful and unique snowflake after all, but it turns out maybe the bacteria are. So how human are we? Again, you see the number kicked around all the time. 90% of the cells in the human body are microbial, not even human. that fluctuates dramatically. But at a genetic level, you have 23,000 genes. We now know there's 10 to 20 million microbial genes in your body. So you are 99.99999% microbial. So you're more microbe than mammal. So again, that's a little bit of a visual of what that means or what that looks like. So again, I'm just going to move along very quickly and skip over all the headlines with regards to microbes are now connected to autism, microbes are now connected to Alzheimer's, microbes are now connected to irritable bowel disease, Crohn's disease, diverticulitis. They're now connected to celiac disease. They're connected to heart disease. They're connected to all autoimmune diseases from A to Z. They're connected to type 2 diabetes, insulin resistance, you name it. There's a microbe connected to it in some way, and there's some lab somewhere in the world trying to make that connection. They've got the correlation on a lot of it now. Now people are just working on causation. So at the moment, the business is actually the business of the microbiome, of the research community, is poised to ask some very important and simple questions. The overarching question right now that's on the mind of literally thousands of microbiologists, bioinformatics, and biological ecologists, medical doctors, nutritionists, it doesn't matter what part of the business you work in, is how do we manipulate the microbes to improve our health? How are microbes distributed over our body? How do our microbes change over time? And how can we translate human and animal models? So much of this work, just like a lot of work in medicine, is done on mice. Not so much pigs anymore, mice. And again, there's a problem connecting those causation and correlation back to humans. So again, microbe is the new black now. So starting in 2000, there was hardly any publications on the gut microbiome. The word microbiome, it's also microbiota, okay? Microbiome just wasn't a hot topic. In 2012, there was almost 1,500 publications. As of last night, that number is over 3,000. 3,000 publications mentioning the word microbiome. If you add microbiota and other kinds of things in there, it goes up dramatically. So there's a significant amount of research effort now being put into the microbiome, and you see the NIH and other communities in Europe pumping extraordinary amounts of money into it, but it pales in comparison to the amount of money being spent on, say, something like cancer. But that's all going to change probably the next 12 to 24 months. Congress at this very moment, next week, are holding meetings on trying to decide whether or not they want to shift a bunch of NIH funding to the microbiome. So that's probably going to happen here in the next year or two. So what's been worked on for the last 10 or 15 years is basically just trying to get a basic idea who's in there. Who's in there, where are they at, not necessarily what they're doing, but who's in there. So projects like the Human Microbiome Project, which was a $100 million NIH effort that was published 2 or 3 years ago, is a consortium of 80 scientists, or 200 scientists from 80 universities around the world, trying to get basically a picture of what's going on with the human body. So looking at things like hair, looking at things like palms, the bottom of your feet, the vagina, looking at the gastrointestinal tract, underneath your armpits, on top of your shoulder, behind your neck, everywhere you could possibly run a swab across a human body, they started to get a map of the different groups of bacteria. And as you can see, these colors are different groups of bacteria, things like Firmicutes and Bacteroides and Proteobacteria and things like that. You can see quite a bit of variation on the human body. The difference between the bacteria in the human gut and the human vagina, they are more different than the bacteria in the ocean, and that's found on land. So it's that different, those two different sites. It's controlled by moisture, pH, and nutrient supply. So where do we get our microbes from? This is a real simple little kind of illustration of what a baby looks like 20 minutes after birth, the difference between a vaginal baby and a C-section baby. More or less, the baby's sterile when it passes through the vaginal canal. There are some microbes in there, but it's more or less sterile. So on the left is the mother that delivered a baby vaginally, okay? And you can see in this particular case, this group of bacteria right here is mainly lactobacillus. But when that baby passes through that birth canal, it's inoculated with the mother's bacteria. And that's been going on for millions and millions and millions of years. And so if we look at that baby 20 minutes after it's born, we still see these group of lactobacillus scattered on various parts of the baby, not so much in the bum yet because it hasn't had a bowel movement. But if we look at the mother on the right side, this is a baby that's been delivered by C-section. Both my children were delivered by C-section. So the baby's basically inoculated with skin bacteria from the mother's abdomen. You'll see there is no red on this baby. There are none of those groups of bacteria. The baby did not receive that seeding population of that pioneer group of bacteria from the mother. And this is kind of what this looks like in a principal coordinates analysis plot. Basically what this means is you guys have seen those little things you shake up that have snow in them, okay? You can turn them, look at them in 3-dimension. So this is basically principal coordinates analysis where basically the closer the dots are together, the more similar they are to one another. So these are actually samples from a baby, that very baby that was taken C-section on the previous slide. And you can see the mother's swabs from the mother's mouth all grouped together. So the bacteria in the mother's mouth are up in here. If we look at the mother's vagina, this is red down here, and this is the baby that was born vaginally that has skin swabs off of it. You can see that the skin bacteria now on the baby look just like the mother's vagina. And so far, so good. This is what it's supposed to look like. And then what happens is this is the mother's skin from the C-section, okay? And this is the C-section baby's skin. So it looks just like the mother. So no vaginal bacteria on a C-section baby. Both my children, again, were delivered C-section, breastfed for less than 6 months, and both of them received broad-spectrum antibiotics before they were 6 months of age. My son, who's 20 now, is fine. My daughter became a type 1 diabetic at age 2. We now know that C-section, lack of breastfeeding, broad-spectrum antibiotics was the 3 things that probably converged on one another to deliver her an autoimmune disease. So 1 out of 3 children in the United States is now born C-section. When my son was born in 1995, that rate was less than 20%. Private hospitals in China, it's as much as 80%. It is probably the greatest public health problem on the planet outside of infectious diseases. And it's also going completely ignored by every administration in the last couple of decades. We can also understand how the microbiome develops in infants. This is a study done on a child where they literally sampled the baby every day or every other few days for a couple of 100 days of its life. And so what we see, again, this is one of those principal coordinate plots where the closer the dots are together, the more similar they are. So this is the baby through time. And so as the baby gets older, the bacteria in the baby's gut, these are stool samples, the baby's bacteria start to change to where eventually, right around 2 years of life, the stool sample in the baby starts to look like the mother's stool sample, okay? So this is a formula. This is basically when formula was introduced to the baby, and this is solid food. And what happens with the baby's bacteria, it gets more diverse over time. So what's important here is throughout human evolution, we all were pretty much breastfed for 2 to 2.5 years when we look at hunter-gatherer groups around the world. Sometimes it's a little less. Sometimes it's as much as 7 years. So when we break that, which we broke it here in this baby as well, we're changing the nutrient supply to the bacteria. So not only is C-sections changing the bacteria on the children, which ultimately affect the bacteria in its gut, we now know from large studies now that cesarean babies have more issues than vaginal babies as it pertains to the microbiota, but we also know the cessation or the dropping off of breast milk has a major impact as well. But usually should be around 2 to 2.5 years, the baby should have a gut bacteria, a gut microbiome that looks very much like the mother's. We can also look at children across cultures as well. So this is a plot showing the actual diversity of the bacteria in the baby's gut compared to adults. And so this is these children from Malawi, these Amerindians from Venezuela, and then the blue dots are children in the U.S. Pretty much the same pattern. So as the children get older, this is years down below, right around that 2-year point, they start to look like adults. So again, they look less crazy, less chaotic, and look a little bit more uniformed and a lot more like mom and dad. And so, although converging on different time points, so even though you can look at a child and see its bacteria changing over time between 0 and 2 to 2.5 years, what happens when those children are adults is something very different. So in this plot over here, these are all adults. The blue dots are Americans. The red are Malawi and Amerindians. Again, this is a 3-dimensional plot. You can imagine turning it. So Amerindians in South America look more like people in Malawi than they do like people in the United States. So even though we all start off more or less vaginally with a certain set of bacteria, we end up at a very different place. And the question is why? Why do Americans look different than people in South America and South America from basically Central and East Africa? And so is it geography? Is it diet? Is it disease? Is it hygiene? Is it antibiotics? Is it the BBC? What is it? And so, but the Human Microbiome Project only covers the start of the diversity. So that's the same plot, okay? This area over here. So this was the Human Microbiome Project. This was a $100 million effort that was finished a few years ago where they looked at about 250 people. It cost $100 million. to sequence the bacteria in extreme detail on 250 people. They did a lot of other cool stuff as well. But the Human Microbiome Project just covered the front end of that diversity and what's going on in the population. There's so much more going on out there, but we don't know what's going on, what's impacting it, what's affecting it, and what direction we should be pushing it. So one of the things that we did starting about 2 years ago, we started the American Gut Project. Has anybody heard of the American Gut Project in here by chance? Is anybody here signed up for the American Gut Project? Oh, good. So this was basically the way it works is the NIH spent $100 million looking at 250 people in detail. 2 or 3 years ago, it was very difficult if you wanted to know what bacteria you had in your body. You could take a poop sample and send it to a lab. It's very expensive, $400, $500, $600. But if it was cheaper, you just didn't get a lot of information. So I teamed up with Rob Knight at the University of Colorado. Rob is a Howard Hughes Medical Institute researcher, 36 years old, way too smart for his own good, probably won a Nobel Prize before he's 50, one of those people that just really, really hate. You really want to hate because they're so smart. So Rob and I teamed up. We said, hey, what we should do is start a project where anybody in America can sign up, or anybody on the planet can sign up and send us their poo, and we'll tell them what's in it, and we'll build this global database of the diversity of the human gut. So far, the biggest project to date was 250 people. That was 250 people, average age less than 30. They were college students at medical departments. That's not the diversity of America. This is the diversity of America. Tall, short, black, white, ugly, Republican, Democrat. So we said we want to get athletes like Ben. We want to get couch potatoes like David, let's say. He's not a couch potato. And then people like me. So what we did is, the big problem with that are IRBs, Institutional Review Board approval. So we spent about 6 months arguing with the University of Colorado at Boulder that we wanted to do this, and they have a panel of people that sit around and tell you why you shouldn't do it and why they're not going to let you do it for ethical reasons. So we got it approved, and then we launched it on Indigo, which is another project launched about the same time. And in about 60 days, we got a couple thousand people to donate $99. So you donate $99 to the project. We send you a little swab about this long. You do your business. You send it back to us. And then we sequence what's in there and send it back to you. Major problems getting the project started. It's all done volunteer. The $99 goes to buy supplies. There's no full-time employee, but about 35 graduate and postdocs that work on it. Again, people way too smart for their own good that have other stuff to work on. So anyway, we launched the project, got all the kinks worked out. So now we're up to about 13,000 people in the project. We launched it on Indigo, and then we continue the project on fundraisers. So to date, we're the largest crowdsourced, crowd-funded science project in the world. Our target is about 20,000 participants, although we might shoot for 100,000. And what we're finally seeing is the diversity in the gut across the United States, and we're in about 140 other countries in the world we've started to get samples from as well. It's a collaboration. Again, it's all a bunch of do-gooders that are helping out with the project, donating their time and expertise and their labs to help. There's 50 collaborating labs. The Human Microbiome Project was a little bit bigger than this, but the total number of people that are actually paying attention to this are in the hundreds, and they're all volunteering their time and expertise. And the entire project's open source. So the idea is that we collect the samples, we sequence the bacteria, we give you the results back, we put the data into these large public databases. It's de-identified, nobody knows who you are. They don't know who you are, they just know it's poop. And we also do vaginal, we do skin swabs, we do lots of other kinds of things. And then all these really smart people, these postdocs and all these people with way too much energy and smarts and time on their hand can look at the data and look for patterns and look at things that interest them in their lab. And hopefully through this open source effort that we can actually start to learn something about the diversity of the American and human gut for that matter. Again, get involved, but it's not for everybody. This is Daniel and Rob's lab at the University of Colorado. This kid I'm a little worried about. That's like a young Bill Gates or something. Anyway. So again, this little graph was done when I think there was about 3,000 people in the project. And so if you imagine major groups of bacteria, they're like automobiles, Chevrolet, Ford, Toyota. Major groups of bacteria like Firmicutes, Bacteroides, Proteobacteria. So when you look at major groups of bacteria. And within each one of those, just like a Ford, you have a Ford Ranger, a Ford whatever, and a Ford whatever. So you have firmicutes, and underneath firmicutes, you have family, order, class, genus, species. But looking at it from a 50,000-foot view, just the brand, if you will, Firmicutes bacteroides, you see that across 3,000 samples, each one of these little lines is a person. Here's me over here. I mainly had in this sample about 70 or 80% firmicutes. Okay, and then Michael Pollan at one time had about 60% firmicutes. He took some antibiotics, and then his firmicutes dropped. Shannon Ford, who was Miss America 2011, she barely has any firmicutes. She also has celiac disease. And then Michael Pollan took some more antibiotics, and his firmicutes dropped. In 2005, there was an article written in the New York Times Magazine. I remember exactly where I was sitting when I read it. I was in a coffee shop in New Orleans in the Bywater. I was there volunteering, helping gut houses after Hurricane Katrina. The article came out and connected bacteria to obesity. Turns out that people that have a high BMI, 30 or above, heavy, a few extra pounds, have a certain set of bacteria. And then people that are lean have a certain set of bacteria. You can take poop out of the lean person, put it in a germ-free mouse, take poop out of an obese person, put it in a germ-free mouse, feed them the exact same food. The lean microbiota creates a lean mouse, and the obese microbiota creates an obese mouse, holding the food constant. And you can play games with food and stuff like that after that. And so that was the first time the public got a real good dose of microbes that are controlling your weight. And so since then, there's been hundreds of studies. Some of them weren't able to replicate that work. Some of them were. But it's gotten a hell of a lot more interesting since then. But the thing that struck me about it at the time, the argument was that the reason that, yes, ma'am?
I just wanted to get your opinion on fecal transplants and that might be something down the road to help heal people's bacteria in their colon.
Yeah, we're going to talk about that towards the end. That's a great question, and we'll get to it towards the end if that's okay. Okay. What struck me about the article was it was done out of Jeff Gordon's lab out of St. Louis. The argument was that the obese people had a set of bacteria that were really good at extracting calories from plants, from plant material. In this case, it was basically fiber. Again, fiber by definition is something that you can't get any energy from. If you swallow a pencil, for example, God help you. If you swallow a pencil, it ends up in your colon. The bacteria can break down that cellulose. Okay, so by definition, fiber is anything that you can't digest. It's not food for you. It's not for you. You do not possess the enzymes to break it down. It's food for bacteria. So what's striking about the article was it said that the obese people had bacteria that were really efficient at extracting or breaking down or creating short-chain fatty acids like butyrate, propionate, and acetate. And I thought, well, that's exactly what they're supposed to be doing. And it was just really odd to me at the time when I read it. So I wrote a letter to the editor of the New York Times. I was all excited. I got my 63 words published in the New York Times Magazine. And that kind of started me down the road of thinking about maybe we're missing something with regards to the obese microbiome, that maybe we can look to other populations to try to understand maybe that argument may be a little bit off. But anyway, back on we'll talk about that in a minute. On the American Gut Project, again, we're not snowflakes. We're all different. You're different. You're different. I'm very different. You're different. Gretel's different. I'm sure David's different. So we're all different. So if you look at the microbiota, you're 99.9% similar in terms of your genome, one you got from mom and dad, but you can be as much as 90 to 95% different in your microbiota. Are we supposed to be that way, or are we all supposed to be a little bit more similar? So through the American Gut Project, here's the Human Microbiome Project, 250 participants. Each one of these dots represent an individual. And what this goofy little graph represents is that when we just look at a small number of people, these numbers are basically differences. The higher the number, the more divergent your bacteria are. So not until we look at about 3,000 people before we find your microbiome looks like your microbiome. So it takes about 3,000 people before you start getting redundancy. So it turns out, up until we found this out about 6 months ago, the wisdom is that everybody on the planet had a different set of microbiota. Turns out it's not true. You've got a twin out there. And the twins start compounding when you get to about 6 or 7 billion people. So it turns out that we do share, but you don't share it with everybody. But should that number be 1 in 3,000, 1 in 2,000, 1 in 1,000, 1 in 500? Our lives and our diet are so different from one another that it's pushed us all in different directions. But the question is, should we all be a little bit closer together? And what does that closer together look like? Should you have more firmicutes, more bacteroides? Should you have more Prevotella? Should I have more alcohol tonight? I don't know. So what we also find in the American Gut Project, when we look at about, I think this is about 5,000 people, 4,000, no, 3,600 people, we see that there's not much difference in the diversity of bacteria between men and women. So you've got maybe 1,000 bacteria, David's got about 1,000 bacteria. So it's not much difference between us in terms of diversity. There is a lot of variability. There are people out there, males and females, with a lot of diversity, and then there's people out there with hardly any diversity, which is scary. So, but it's basically, it cuts across gender, which is important. Remember that fact when we get towards the end of this. Alpha diversity, again, this is diversity. The age, people in their 20s have a less diverse microbiome than people in their 60s. And why diversity is important is if anybody follows kind of ecology, anybody that actually gardens or pays attention to the landscape, basically Ecology 101 teaches us that diverse ecosystems are more resilient to perturbations, fire, nutrient starvation, moisture, and it works the same with the gut. The human gut is the most diverse ecosystem on the planet. There's more species in that little bit of real estate than you'd find in a square kilometer in the Amazon rainforest, okay? Depending on how you define species. And so we see that as you get older, the gut becomes more diverse, which is interesting, which is why this is, we have people that are in their 70s and 80s now. Diet changes, the diet actually becomes less diverse. This is when a lot of medications start showing up, but we continue to tick upwards over time. And this was unknown in the large population until the American Gut Project, which was made possible by people donating $99. Turns out there's a lot of variability and diversity throughout the year. So even though these little red lines aren't that far apart from one another, basically it's statistically significant. People's bacteria changes throughout the year. The diversity changes. And basically it changes between, again, it's the highest diversity in the winter than it is in the spring. You would think if you're out at the farmer's market or you're running around outside because the snow melted, you're swimming more, spending more time outside, all those microbes, you'd be kind of swallowing them up, you know. But it turns out in the winter, maybe when we're a little bit closer to one another, our diversity goes up. We also eat a lot of crazy food during the winter and drink way too much eggnog. So whatever it is, we're more diverse in the winter. It's the first time this has ever been found out. It's been done by a volunteer project. There's been billions and billions of dollars spent on gut microbiome research, but until we look at thousands and thousands of people, it's a goofy little crowdsourced crowdfunding project made possible by people who are generous enough to donate $99 that we start to see some important patterning here. And this is critically important. This is alcohol frequency. People that never drink have a less diverse microbiome than a daily person that drinks every day or regularly. So the more you drink, the more diverse your microbiota. We didn't break it down by tequila versus craft beers, but people boozing it up seem to have more diverse microbiota. It's not a green light to go drink, but kind of like it. Frequency of alcohol by gender, okay, works across men and women. Doesn't matter if you're a man, woman. Diversity increases with alcohol. Has a little bit to do with undigested sugars, we think, in the alcohol that end up in your colon. It promotes diversity. It's not necessarily diversity of good bacteria. Could be bad bacteria that are blooming up because of the tequila, but diversity goes up overall through the ecosystem. Exercise frequency. David, you'll like this, and Ben too, with no shoes or whatever he was wearing yesterday. People who exercise daily have a more diverse microbiota than people who occasionally exercise. That's not a rocket science. You know, you spend a little bit more time outside. Maybe you're absorbing some more bacteria. Who knows? Maybe it's hormones. Maybe it's who knows what it is. Sleep. This is a good one. People that sleep more, and again, this is looking at thousands of people that when they send in their sample, they have to fill out this questionnaire, which they really hate doing. And how many hours do you sleep? How many this do you eat? You know, do you have a dog in the house? Do you drink well water? Do you use tap water? So people that sleep more have a more diverse microbiota than people that sleep less. Again, this isn't hard to wrap your head around. It makes a lot of sense. Microbially is a good thing. This one is really fascinating to us. One of the things that we do, we ask people to fill out this food questionnaire, basically where they have to track what they eat for a week, and it drives people crazy. If you've ever tried to fill out one of those online things where I had bacon for breakfast, and it's horrifying when you fill those things out. You're like, geez. And so you start filling them out, you just stop eating so you don't have to actually fill the damn thing in. So I think that's how people lose weight on those things. But we ask people to count how many species of plants they eat in a week. The average American, according to the U.S. Department of Agriculture, there's a weird website that has this spreadsheet on it that tracks information like this, and I got to get a life because I look at this thing all the time. The average American eats less than 5 species of plants a week. Tomatoes, French fries, potato chips, things like iceberg lettuce. The average American eats less than 5 species of plants a week. Okay? So we asked people to write down, spices don't count, how many different species of plants you eat in a week. And we found that people that had more than 30 species of plants in a week had a significantly larger diversity of bacteria. Now, that's also not a no-brainer. Nobody had ever seen this pattern, and I came up with the question because I like fiber, and I like fiber's role in health and well-being of bacteria. So if you get up in the morning, you eat 3 bowls of bran cereal and slap yourself on the back because you just got 35 grams of fiber, doesn't matter with regards to diversity. What you need to do is go to the grocery store. Average grocery store, big ones have between 150 and 250 species of plants in there. Average American eats 5. So it's for fun. You guys should do that starting Monday. Just write down species of plants you eat in a week. Shoot for more. But are we modern humans the best people to study? So what's happened for the last 20 or 30 years, 40 years, 50 years, mostly in the last 10 years, people that want to understand the microbiota, let's say you're in St. Louis, you run an ad in the paper, say you want to make $30, come poo in a petri dish for us, and we'll give you $30. It's like donating blood. Everybody that they're analyzing, and everybody that's part of these studies are drinking chlorinated and fluoride water, we're drinking alcohol, we're smoking weed, we're drinking, we're eating too many potato chips. Basically, we're all living a Western lifestyle, and not anybody in this room would argue that our lifestyle and diet today, except for maybe David's over there, resembles anything like our ancestors. I keep picking on David because he's the only one I know here. And I know Gretel. And so the question is, are we going to learn anything by looking at sick people? So when we look at a group of people, like 250 people in the Human Microbiome Project, 250 healthy people, they were defined as college-age students. What do college students do? They smoke weed, they do coke, they drink, they don't sleep. And these are healthy people. And we use that population or that cohort to characterize a healthy microbiome, for God's sakes. Okay? And so every single microbiome study right now that's taking place in the United States or Europe or Australia or South America, every one of these studies is being done on people that are sick. Okay? Even if you look good, you still have a Western gut. Okay? So the question is how much are we actually going to learn? So about 5 years ago, I said, you know what? What we ought to do is we should look at ancestral populations. We should look at people that haven't logged on, so to speak, to Coca-Cola, cooking oil, antibiotics, Justin Bieber, whatever, okay? They haven't been compromised or ruined in any way. So we started off about 3 or 4 years ago working in South Africa, basically not here, but in Botswana and Namibia, the famous San Bushmen. Okay, so we went down there, looked around a little bit. And of course, the San Bushmen, if anybody knows, have basically all been herded up and put on reservations, just like we did to our Native Americans here. And they're being delivered government subsidies every month, and it's against the law for them to hunt. Thank you, De Beers Diamond Company. Okay, so if you're a San Bushman, the Gods Must Be Crazy fame, you will be thrown in jail if you kill an animal. You'll be thrown in jail if you kill an animal. So I spent a lot of time with the San Bushmen in South Africa and Namibia and places like that. And again, they're compromised just like you and I. I sit down with some San Bushmen who run up and put on the skins because they think I'm a tourist, but the guy had corn flakes for breakfast and a beer for lunch and some corn oil for dinner. So completely compromised population. I did spend a little time in Namibia. We found a population up in the mountains that were part-time hunter-gatherers. Took a couple days in to hike in and find them. We magically found them. I was the second white man they'd ever seen. But they did have access to goats and cows. So Tanzania, the Hadzabe. Have people heard of the Hadza before? The Hadzabe? They've been studied pretty intensively since the early 1900s and pretty heavy duty since the 1970s. Frank Marlowe's group out of Cambridge has been working there literally 3 or 4 times a year for the last 15 years. They're the most studied hunter-gatherer group on the planet. And they're basically located in Tanzania, which is East Africa. Here's Kenya, where's Nairobi. Ebola is way over here, by the way. All the emails are like, you got the Ebola yet? No. It's like 4,000 miles away. So anyway, so we spent about 6 months to a year getting the permits, which was a pain in the ass. Can I say ass? David, is that all right? It's a pain in the ass. We got the permits. We put together a collaboration of a who's who in microbiology. Rob Knight at the University of Colorado, that guy that's way too smart for his own good will probably win a Nobel Prize. Marty Blazer at New York University, who was the chair of the medical department there, pioneer in microbiome work, just wrote the book Missing Microbes. Highly recommended if anybody wants to get it. Missing Microbes. It's the best microbe book out there until mine comes out. I need the $2 so y'all can buy it. I appreciate it. Marty Blazer, his wife, Maria Gloria Dominguez Bello. There's some other names in there, but I think that's it. She's from Puerto Rico. She's Puerto Rican. She's a lot of fun. She's a microbiologist. She's on the project as well. Gregor Reed, who's the world's leading authority on probiotics and the vaginal microbiome. He's on the project. Jose Clemente from Mount Sinai, New York. Sam Smits from Stanford, Justin Sonnenberg from Stanford, and 3 Tanzanian researchers. So we put together this huge team, Jack Gilbert from the University of Chicago, and all the postdocs and graduate students in those labs. So if you put them all in a room, you would need a room 2 or 3 times this big. But somebody had to go get the sample. So we put the team together, got the permits, and off to Tanzania I went on August 7th, 2013. I was on the ground in Tanzania a month before I finally saw my first Hadza. So I tried to go to the field, get a flat tire, try to go to the field, the truck would break down. So they're basically 8 hours away from anywhere useful, i.e. a gas station or anything like that. So the Hadza, there's about 1,200 Hadza left. Of those 1,200, there's about 100 to 200 of them that are still full-time hunter-gatherers. What that means is they get up in the morning, they go hunting, the men go hunting, they hunt with poison arrows, and the women get up in the morning, they go dig tubers and gather other kinds of things as well. So they're in an area, Lake Eyasi. I'm going to go back. So this is Tanzania. This is Lake Eyasi right here. Here's Arusha. Here's Mount Kilimanjaro right here. And despite what the cultural tourism officer from Kenya says, Mount Kilimanjaro is not in Kenya. It's in Tanzania. So Lake Eyasi right here, again, so this is Lake Eyasi. It's a 50-kilometer long lake. The Hadza in the 1950s had a huge area, massive area. They've been shrunk down to a very small area now, mainly because of agro-pastoralists, mainly pastoralists, the Datoga, which look a lot like the Maasai. So anyway, this is Lake Eyasi in the distance. This is the Hadza guy here. So what we were interested in are the Hadza that are still hunting and gathering. There's a bunch of Hadza that are now on the tourist circuit. There's a place called Mongola, Barazani. Has anybody ever been there? Okay. There's a place where safari vehicles roll in every day, about 5,000 of them a year. Roll in, the Hadza run out, they put on their baboon skins, they dance around, and they put their hand out for money. Okay? So these Hadza are part-time hunter-gatherers, but they are now part of a cash economy. So they got access to alcohol, antibiotics, everything. So terrible, but useful from a microbiology standpoint. So we have Hadza that have passed through that epidemiological transition, just like the rest of us have. They got access to almost everything you and I have, okay? So we can study them, and then we can study Hadza that are partially acculturated, and then we can study Hadza that are still hunting and gathering and living a little bit farther out, if you will. And so that represents literally 10,000 years of human history, agriculture, antibiotics, indoor plumbing, Justin Bieber, okay? And cell phones and all the other crap that go with all that. So in one population, one genetically population, we can monitor the bacteria across these epidemiological transitions. And why nobody had done this before, I could never figure it out. It's very, very hard to do. And most microbiologists prefer to work in labs. It's very hard to work in rural settings. My colleague, Maria Gloria Dominguez Bello, at NYU, she works with Amerindian populations in South America. Field work in East Africa is hard, very hard. The first 3 months we were there, we had 50 flats. The vehicle broke down 10 times. There's no water. It just never ends. You have to bribe the cops nonstop. I mean, it never ends. And so it's hard. So we pushed through and we started this project last summer, working with the Hadza. And again, the Hadza still hunt. And why they're very interesting is they still hunt the same animals that humans have hunted for millions of years. They live ground central for human evolution, for the evolution of our genus Homo. Unless you guys follow Lee Berger in South Africa, South Africa has given East Africa a run for its money now on the cradle of humankind, but you can throw a rock and hit Olduvai Gorge from here. Okay, so we have Homo erectus, we have Zinjanthropus boisei, we have everything in the area. So the Hadza literally hunt the same animals minus the megafauna that humans have been hunting for millions of years. They gather the same plants, dig the same tubers, are covered in the same soil, drink the same water that humans have been evolving with for millions of years. So from a microbial standpoint, I don't care if he has an LA Lakers t-shirt on, okay? Has nothing to do with his or her microbes. I don't care if they have access to short pants and he's got a cell phone. The babies are still born in the dirt in a hut. They do not have or have limited access to antibiotics, limited access to this, limited access to that. They still live in an ancestral environment. I'm not arguing that we should all eat an ancestral diet, or that you'd have so much of this or so much of that. They live in the microbial community which gave rise to our genus Homo. They are the most perfect referent population in the world for understanding what microbes we might or should be carrying, okay? More so than, let's say, Yanomamo in South America or Arctic hunters, okay? Those are environments that humans populated very, very late, and the microbial communities are very, very different. So the microbes that trained our immune systems, the ones that turn off and on helper cells, are right here in Tanzania. And so here's a couple of guys killed some baboons. We were here for this zebra kill. Taryn was here, had her first zebra about a month and a half ago. There's Taryn in the background. This is an impala. Again, these are baobab fruits. You guys are all familiar with the baobab tree from Lion King. Baobab, the Hadza consume 750 different species of plants and animals. I think it might be as much as 800. 90% of those are birds. Okay, the baobab fruit, okay, which is this chalky little thing on the inside with a nut on the inside, delivers more calories than any other species, plant or animal, in their basically landscape. So lots of berries. Women dig tubers. Here's women roasting some tubers. I'm just going to gloss over a couple of quick things. There's a man climbing a baobab tree. Here's some wild honey. During the wet season, like January and February, sometimes in April, honey could make 60 to 80% of the calories for weeks on end. When I was there in January and February, I was there 2 weeks on the ground. We didn't have a single piece of meat. They were all vegetarians. Okay, and that happens every wet season. So when the rains come, here come the flowers. They don't hunt as much. When the rains come, or during the dry season, the water holes are really small, so the game is really predictable, so they kill lots of game. But when the rains come, the water's everywhere, the animals spread, they don't care. They would rather eat honey all day. And so if you interview 100 Hadza men, ask them their favorite food, more than 50% will say honey. If you interview 100 Hadza women and ask them their favorite food, more than 50% will say tubers. There's a massive sexual division of labor in the Hadza. The men hunt and gather honey. The women dig roots and gather berries. They don't even sit together. Okay? If you go to a camp, the men are over there, the women are over there. They're completely separate all the time. Sexual division of labor. So the thing we wanted to know from the Hadza was, and they still live in traditional houses, the things we wanted to know from the Hadza was, because there's such an extreme change in weather there, you have 6 months of rain and then dry. And that's been going on for 20 million years in East Africa. Is it important that humans have been following a seasonal rainfall pattern and the resources change season to season? A lot of meat in the dry season, a lot of honey and berries, and lots of fiber in the wet season. Has that shaped or basically modulated our microbiota in any way? So nobody had ever asked. We see this in the American gut data where it changes, but when we started this project 2 years ago, does seasonality affect the microbiome? And why this is interesting is if you go to your local health food store or talk to any smart people around you, what bacteria should I have? Should I take this probiotic? Should I have Lactobacillus acidophilus? Should I have Bifidobacterium? What bacteria should I have? And that's a complex question. It depends on how old you are and your disease state and so on and so forth. But it turns out the question is does seasonality affect our microbiome? Almost like circadian rhythms, should we have different microbiota at different times of the year based on changing resources over the last millions of years of human evolution? So that was our primary research question. Our hypothesis was, did it change? And then we had a list of 100 other research questions as well. And so basically we asked the Hadza to participate. We read to them. We get informed consent. Most of them can't write, so we get a thumbprint. And if they agree to participate in the study, we get a small vial of poo from them, and we store it in liquid nitrogen in the field. Again, a lot of people don't do these projects in rural places because you can't get liquid nitrogen. And so it's just everything's hard when you work in East Africa. Well, they call me Dr. Mavi, which is translates Swahili for shit, which was fine. And shit in Hadzabe, there's only like 3 or 4 people in the world who speak Hadzabe. The word shit in Hadzabe is hichi. But anyway, they used to call me, oh, here comes Dr. Mavi. And then they just drop the doctor. They just say Mavi. So like, they don't mean anything by it, but it hurts a little bit every time. So But anyway, here's that rainfall I was talking about. Now, this rainfall pattern and resource availability has characterized human evolution ever since we were knuckle dragging. I mean, it's been around forever, and this is important. This is really, really important. So if we're going to sit around and talk about what an optimal or balanced microbiome looks like in the West, we better decide if this is important to us. Okay, it may condition what probiotics we take or don't take. Right now, nobody considered it. Nobody does. And this is a multi-billion-dollar business, and nobody's thinking about it. So anyway, there's the rainfall. So right now, it's dry as a bone there. I'm all excited because the Hods are killing lots of animals. Taryn and I'll be back there in November, and hopefully I'll get to taste my first giraffe. They also kill lions. They kill giraffe. They kill kudu, impala, warthog. They'll kill everything. They can't kill elephants because their poison arrows aren't strong enough to bring them down. So anyway, this is Rob Knight from the University of Colorado. At this particular moment, he looks like an elderly woman shopping for real estate in Florida. Scary, smart guy. This guy invented all the analytical tools that all the biologists and everybody in the world uses to analyze the sequencing data. And there he is, 36 years old from New Zealand. Just moved to the University of Colorado San Diego on January 1st. So in addition to taking poo samples, they laugh at us. A lot of them, to answer your question, a lot of them don't want to participate. It's difficult. It's very, very difficult to do. Most of them are a little iffy about it. But in addition to that, we're also looking at skin samples. We're looking at swabbing their hands. We started taking spit samples and oral samples recently. We're moving towards blood, which I'm a little nervous about. We don't think they'll want to do it, and we might not end up doing it. But we're looking for correlations between their bacteria in their gut and what's going on in their blood as well. We do groin samples. We also do a significant amount of environmental sampling. We sample the water. There's cows in their environment, so we're interested in whether or not the bacteria in the cows, that the shit ends up in the water and then the Hods that drink the water, if that's translating through them as well. Then we also sample animals. This is a zebra kill about a month and a half ago, so we take stool samples from the zebra as well. So if you're going to understand the human gut, you have to understand where you get your microbes. So this is the largest wildlife microbiota project in the world, too, just by default, because the Hods kill lots of animals. So we get to sample the animals. And Dick and zebra, they killed a lion recently, but we weren't there for the lions. We didn't get to sample that. So we're looking at what was called the meta-community. All the bacteria around. A cubic meter of air in this room right now has millions of bacteria floating in it. Every time you take a breath, you're taking in millions of bacteria. If you kiss somebody, that number goes into the billions if you're smooching really deep. And so we're interested in all the bacteria. We're interested in how the bacteria changes in the water throughout the year as well. Does the bacteria change seasonally in the water? Does it change seasonally in the zebra? Does it change seasonally in the dick, the giraffe, the warthogs? We also just launched a project with Alex Peel and Fiona Stewart on a chimpanzee project in southern Tanzania. So right now there's some guys collecting chimpanzee poo every week for us so we can see if there's seasonal changes in chimpanzees as well. We've also are launching a project with a guy named Dr. Foley at Tirangiri. We're looking at changes in the bacteria in elephants throughout the year as well. So it turns out that probably the bacteria change seasonally in basically every little critter on Earth. Just nobody ever asked the question. And the big question is it important? So lots of environmental sampling. But most importantly, still connected to the larger microbial meta community that made us who we are, we want to know what interactions get you to exchange microbes with your environment. Obviously, drinking water, you'll absorb those bacteria, but we want to know what we used to do as a population, everybody in this room, our ancestors, how we used to acquire microbes, and maybe how we can rewild our bodies. And then we're all not going to go out and go zebra hunting or drink water with cow shit in it, okay? But we want to understand how that larger meta community basically delivered. It's the original social network, if you will, the microbial social network. By being in this building, we've shut it off. We're no longer outside anymore. We shut it off. So maybe we want to open a few more windows or spend a little bit more time outside or maybe do a little bit more gardening. But the Hods are intimately connected. And it seems really simple, but until you sit around underneath the baobab tree and hang out with some Hods, this little girl actually bit the head off this bird about 2 seconds after I took that photograph. Okay, and all the bacteria on the tip of that bird's head ended up in her mouth, and obviously all the bacteria on her hand, and she touches her face, she touches her little brother, her mother touches her. So that movement of microbes between these little islands that we call people is what we've basically lost in the West. So again, we no longer go out and do this much anymore, okay? Here's some Hadza butchering a big kudu. And then one of the things they do, which was fascinating, first time I saw this, I literally sat down. I couldn't believe I saw it. They gutted a big kudu, and then they took the stomach, and they hung the stomach in a tree, and they slid it open, and all the stomach material came out, which is poorly fermented, grass and so on and so forth. And then after they got through butchering the animal, their hands are covered in blood and feces and everything. They went over and stuck their hands in the stomach contents because it's acidic, and it helped. I don't know if you guys have ever had blood on your hand. It's really sticky, and so it helps clean the blood off their hand. So the extraordinary exchange of microbes between a human and another mammal, it just never dawned on me until you actually see it. So this has been going on for millions of years. This is the stomach contents. And then the first thing he does is he does this, and then he does this, and then he does this, and then he touches his friend or touches his kid. So those microbes are moving all the time through the act of butchering. And so what do? We study the bacteria in the stomach contents, and we can track, just like a fingerprint in a FBI fingerprint book, we can track the exact bacteria from that animal through the gut of the Hadza and see whether or not it persists or stays. And so anyway, this is huge for human microbe symbiosis throughout human evolution. Of course, everything's now nicely wrapped at the grocery store. This guy is eating, this is a raw stomach. Hasn't taken much time to clean the blood off of here. Okay, this is a guy named Saidi. He's one of our star Hadza. How many kids does Saidi have? 6, 5, 6 kids. So we're not only studying individuals, we're looking at mom, firstborn, secondborn, thirdborn, fifthborn, all the way down. Then we're looking at grandmother, great-grandmother. So we're able to look at succession of bacteria across multiple generations. And grandmother is sitting on the ground playing with the baby, just like they've done for millions of years. What do we do with grandmother now? She's in an assisted living facility, okay? So she still exchanges microbes with the 1-year-old. She still exchanges microbes with the son-in-law. And again, so he's consuming extraordinary, this isn't cooked, okay? This isn't cooked at all. The first time I saw him eat raw colon, I had to sit down again too. They offered it to me, I'm like, good. So they took the colon out of an impala, cut it up sushi style, and ate it. And I just sat there, couldn't believe it. So the extraordinary exchange of microbes from eating raw colon, eating stomach, blood, they're covered in dirt, they don't bathe. This is the environment which gave rise to our symbiosis with bacteria. All right, here's the other thing. So last year we collected about 500 samples. We just sequenced the data. We're just getting ready to publish it, so we don't talk about a lot of it. So the first 500 samples, we've now collected another 1,000 or 1,500 samples. We now know the Hadza have the most diverse gut ecosystem in the world of any population that's ever been looked at.
I was wondering if you might talk then the health of the Hadza, how they compare to the average.
Good question. Let me run through this real quick. So we see, these are what are called operational taxonomic units. So that's a fancy word for species, okay? So basically and again, you can go down to individual strain level and that number goes to 40,000, okay? So basically the average American has around 1,000 species of bacteria, or OTUs, operational taxonomic units. When we look at, I don't even know if I spelled that right, when we look at people in Malawi, it's a little bit more. Amerindians Indians in Venezuela that Maria Gloria works with, and then the Hadza, okay? So the Hadza have almost basically twice the diversity of gut bacteria than the average person in this room. And the question is that important? And to your question, Gretel, people always say, well, the Hadza don't live very long, or they don't live long enough to develop diseases. You guys have heard all the arguments and stuff. The average life expectancy of a Hadza is 35 years, okay? It's not because of diet. It's because of high childhood mortality. So let's say you and I had a baby. I'm 30. You're 30. So you and I are 60, okay? If we had a baby, so that's 60. Our average age is 30. If the baby died at birth, you'd have to divide 3 into 60. So now average age life expectancy is 20, okay? That's how that number is calculated, all right? So 1 out of 5 children die before they're age 5. It's malaria, which I got, pneumonia, people fall out of trees, simple infections kill people, childbirth kills people. It has nothing to do with diet. If you get past 5, 10, we've got 80-year-old women in our study, 85-year-old people in our study. So it's a rough life. When you look at somebody and you ask them how old they are, they don't know, for starters, so it's difficult to assess age. But a 40-year-old looks like a 60-year-old. A 25-year-old looks like a 50-year-old. It's rough living outside every day. Simple infections can kill you. So the health of the Hadza, there hasn't been any large medical studies. Work's been done in the 70s and 80s, but there's little to no cancer. There's no autoimmune disease. There's no diabetes. There is some alcoholism now. You start to see a little bit of obesity. The average BMI of the Hadza is 20 to 21. It doesn't change at all. They'll gorge on honey and eat 10, 15,000 calories of sugar in a week. They don't gain any weight. So it's 20 to 21. They're thinner than Ben. Ben's sick in shape anyway. But they're all very thin.
How are their teeth? Are their teeth quite straight? Do they have room for all their teeth?
Again, I don't do teeth, but it's highly variable. Some of the people have beautiful teeth, and they're 40 years old. Just beautiful teeth. And then a 25-year-old guy's got some crazy teeth. I don't think there's a lot of cavities, again, but I don't do teeth. Some of the teeth are messed up, we think, from the water as well, discoloring from the water, and then tobacco use. They smoke marijuana every day as well, too, by the way. They trade for it. But I couldn't tell you about the teeth. But we're stunned by the variability. You look at one family, beautiful teeth in one sibling, pretty brown and nasty in another. It's all over the place. And what they'll do is they make these little toothbrushes out of these sticks, and they'll sit there and do their thing like this all day. And I think it's the guys who don't do it, they have some funky teeth. The guys that do it have really nice racks. Or not racks. It's a good question. I wish somebody would, if somebody wanted to join the project and study teeth, we could add it to the IRB. So if there's a dentist in the room that wants to pay their own way, come on. Did you have a question or somebody else?
Well, I had a question.
Yes, ma'am.
I was like, how old were some of the elders that you live with?
Yeah, you see a lot of 60, 70, 80, yeah, but it's hard to assess age. So we work with translators and stuff, and then people they don't know when they're born. So we're plus or minus probably 10 years on the older people, plus or minus 2 or 3 on the kids. So again, Hadza are very diverse. Here's the other thing that's absolutely fascinating. Prevotella, which is a bacteria that's talked a lot about in the press in the United States. There's only 2 identified species of Prevotella. So among this crazy diversity that they have, these are all the different species of Prevotella that the Hadza have. Okay, 20. We've only identified 2 in the entire western gut. So the question is this important? Should we be carrying more Prevotella? Prevotella has also been associated with, correlated with things like autism, rheumatoid arthritis, a lot of inflammatory diseases as well. So Prevotella may not be a good thing, and we'll talk about that again in a minute. But the Hadza have this extraordinary diversity of Prevotella. So should we be paying attention to this? Yes? Yeah, 16S and 18S.
Are you getting down to the species with the 16S?
Yes, we can, but not in this. We don't. Depends on what we're doing with it. So yeah, we can get down to species. And Rob's labs also developed some other stuff too, where instead of 97% matching, we're doing 100% matching as well. So we do 16S, 18S. We're also looking at resistant genes as well. So we're looking at the resistome. We're also looking at the virome. So a couple of different things. But this is 16S. So here was the original research question. Do the bacteria in the Hadza change between wet and dry season? Okay, so mid-dry season, that's the blue ones, eating lots and lots of animals, very little honey, no berries because there's no rain. And then this is the same group of people 4 months later. Same group of people 4 months later. They all shift. So when the resources shift. So the question is, if you're in the West, do you want microbes that look like this or microbes that look like that? Should I take this probiotic or should I take that probiotic? And the question is the gut is a moving target. There is no stable state. It's moving, okay? It's fickle. It's shape-shifting, okay? This is the first time this has ever been identified in a human cohort anywhere in the world when we did this. And so this is huge for us. We don't know what it means yet, but we kind of do. It's going to change the way we think about what an optimal microbiome is. It also changes across gender. It doesn't matter. On average, men seem to have more diverse microbiomes as women. Okay? If you remember that American gut slide, men and women are the same. If you look at Americans, men and women have the same diversity. But for whatever reason, men have a greater diversity of bacteria than women. And has it been that way for millions of years, and is it important? Okay? Well, we don't know yet, but it works across gender. This is interesting. I apologize for the small print. We also sampled their hands. This is 157 right hands. We look at the bacteria on a bunch of hands. They all look about the same. They cluster together. But what happens is this is an individual that killed an impala, and before he touched the impala, I swabbed his hand, and so he looks just like everybody else, and then 1 hour after he butchered the impala, that's what his bacteria on his hand looks like, okay? And then he touches his head or whatever. And so now what we're doing is time series where we'll follow the same individual, we'll sample them every hour for 5 days, and then see how long it takes for it to come back, and then we'll sample everybody in the family to see whether or not the bacteria on his hand end up on Junior's head, or on Mama's arm, or on Sally's leg, or whatever it may be. So where we get our bacteria, how often we get them is critical. And so we've cut off that continual supply again in the West, whereas this continual supply has always been there for us throughout human evolution. Here's another one. Again, it's just real simple. This is 1 hour after butchering an impala. This guy way over here, of course, it changed. This is another guy who butchered a kudu. His hand looks very different over here. This is a hand after harvesting honey. They climb up in the tree and they get the honey. Didn't really change the bacteria. So animals are a significant source of bacteria for the human body, okay? And then we also swab the berries and the roots and stuff, and you see similar patterns with those, but there is no pattern with honey. I'll start moving along here real quickly. This is diversity of bacteria through age, through a first group of people. Again, it doesn't matter if you're 2 years old or 50 years old. Males tend to have more diverse bacteria than women. Women are the line here. Lots of variability, though. Here's another one, bifidobacterium. You guys all see the probiotic bifidobacterium. You got to have it. Can't live without it. Activia wouldn't be in business without it. Okay, bifidobacterium is the most dominant bacteria in a breastfed child's gut. Okay, it's also, again, one of the primary probiotics out there. The Hadza, they don't, of course, keep animals, so there's no milk after breastfeeding. So once the children are weaned, here's what happens to bifidobacterium. So bifidobacterium starts to change throughout the life of the child. After they're weaned around 1.5 to 2.5 years, it drops out. There are no bifidobacterium in adult Hadzas. It doesn't exist, and neither does lactobacillus. So the two dominant probiotics on the market in the West today are not even part of the consortium of what I would call our ancestral gut. And also, there are other species that drop with bifidobacterium. When bifidobacterium drops out, there's a co-occurring other bacteria that drop with it. The number one genus of bacteria in the American gut is a group of bacteria called bacteroids. This is your Ford Ranger, if you will. It's everywhere. Drives you crazy, okay? It's everywhere. It's the dominant bacteria. The other one in the American gut, or I call global gut, is Ruminococcus, and the other one's Prevotella, okay? So bacteroids dominate the American gut. Hadza don't have any. They don't have any. The number one explanation in the literature for why Americans have bacteroids is we have a high-fat, high-sugar diet, so that's why bacteroids are there, okay? So I tested that on myself. So I was living in New Orleans last year, and then I drove out to Terlingua to West Texas. Anybody know where Big Bend National Park is? Okay? So Gretel's been there. She's sat around my fire. So I drove from New Orleans in my little smart car. I drove all the way out to West Texas. And in New Orleans, I had a high-fiber diet. Every morning, I'd eat leeks, onion, garlic for breakfast. I was eating lentils, and I was eating 20, 30 species of plants a week. I went out to Terlingua to finish writing a book that I need you all to buy. Again, need the $2. I went out to Terlingua to finish writing a book, and so every meal I ate at a bar, okay? And I drank tequila, and I ate steak, and I drank tequila, and I ate a hamburger, whatever. And basically, I went to a no-fiber diet. So within 10 days, now I apologize for how hard this is to read here, but this red bar right here, okay, this red bar is my bacteroids. Just like every other American. After I went to the bar and started eating every meal there, my bacteroids shot through the roof. But before I got there, my high-fiber diet, but I also ate steak every day in New Orleans. I ate bacon every day for breakfast, okay? So the only difference between my gut was, in New Orleans, I had high-fat, high-protein diet, but lots of plants, okay? No bacteroids. What do the Hadza have? High-fat, high-protein diet, lots of plants, okay? So when I cut the plants out, but kept the fat and the protein, my bacteroids shot to the roof. What happens is when you ferment fiber, you create short-chain fatty acids. So you make your colon more acidic. I also measured the pH in my stool. So what happens is there's a body of research that suggests, this is basically Larry Frent's group out of Europe, that shows that as you ferment, your colon becomes more acidic. An onion's or a lemon's more acidic than an onion. So it became very acidic, okay? Turns out bacteroids don't like acidity. Okay? So the explosion, my hypothesis is the explosion in bacteroids in the Western gut is not because we eat too much fat and protein and maybe sugar, it's because we don't eat enough plants. So we have the most alkaline guts on the planet. So what happens is when you make the colon more acidic, that's like turning up the heat in the room, okay? When you turn the heat up in the room, everybody wants to leave. Or if I talk for another 30 minutes, everybody wants to leave. So anyway, so the point is when you make the colon really acidic, it blunts down other bacteria. They don't like it. They don't grow very well, if you will. And so I think the reason is because we eat less than 5 species of plants a week. The average American eats less than 18 grams of fiber a day. The average Hadza, 1-year-old, eats between 50 and 200 grams of fiber a day. Average American eats 18. So they have super acidic bodies. They have super acidic colons, which select for a certain group of bacteria that not only blunts bacteroids, but also blunts pathogenic bacteria as well. So from a 50,000-foot view, if they put me in charge of the human food supply in America for one day, which they're not going to do, and they shouldn't, but if I was Michelle Obama for a day and I had complete control of Health and Human Resources, the U.S. Department of Agriculture, and I could make one chart for the new My Plate or the My Pyramid or whatever it is called these days, whatever, I would increase the diversity and quantity of dietary fiber. You can talk about all the other red herrings all day long, but if you're talking about microbes, okay, if you're talking about microbes, you're talking about fiber. So what we need to do is increase the acidity of our colons, and there's only one way to do it. So anyway, so that was my Terlingua gut versus my New Orleans gut. And then I had a huge spike in Prevotella when I was out there as well because I was eating granola every day. By the way, Paraprevotella and Prevotella are not necessarily a good thing. If you eat a lot of whole grains, you probably have a lot of Prevotella. So this is really cool. I'm almost done too, folks. So anyway, when we look at so this is a group, this yellow out here is a bunch of Americans again. So they all look similar. Their guts look similar. These are some people in Malawi and Venezuela. They look similar. Here's some people in Italy. Who cares? The blue dots are Hadza. The red dots are vervet monkeys. The Hadza look more like vervet monkeys than they do like Americans. Wrap your head around that for a minute. I have a plot, but I don't have it with me. It's the same with chimpanzees. We look more like chimps, the Hadza, and vervet monkeys than they look like Americans. Interestingly, the Hadza look more like Amerindians in South America than they do Americans. So there's some ancestral depth there, if you will, okay, between Amerindians and Hadza that have been lost in the West. Okay, this is interesting to us. This particular small group, this was a wet season group. Just threw it in there. Not as much meat. Doesn't change with dry or wet. So when you're looking at 1,000, OTUs or 1,500 OTUs, it's not going to move it that much. So we looked at it both ways. This is the only one I have. It'll probably move around a little bit, but it's not going to move over here. And but it does the same with chimpanzees. We had a small chimpanzee data set, and so we're now hopefully have 200 more samples by the end of the year. But anyway, so kind of makes you wonder a little bit. Real quick, I'm almost done. This is your microbiome on corn. We're working in one camp on January 26th. We've been sampling there, and on January, there was nothing. They were eating honey and roots, no animals or whatever. They got their hands on 20 kilograms of corn. Okay, they traded for it and they traded some honey for it. And so they immediately, when they get corn, they eat it for breakfast, lunch, and dinner. Okay, a hunter-gatherer will sit down. Okay, it's sit-down food. They will sit down if you give them corn, and they won't go anywhere because they love corn. They'll just eat it all day. They eat it for breakfast and make this terrible, terrible meal with it. It's called ugali. You can't put enough honey in it to make it taste good. But so this was a dietary intervention. So here we go. We can look at the effects of corn on a hunter-gatherer. They've been hunting. I was there for 2 weeks. It was pure hunting and gathering, basically a vegetarian diet. And what happened is they started eating corn. This was on the 26th, the 28th, the 30th, the 31st. I actually have data for February 1, 2, and 3, but it's not on here. So their Prevotella shot up, okay, that are associated with rheumatoid arthritis, autism. Also, people with AIDS have higher levels of Prevotella. But Prevotella in this particular case, which is another argument I've been trying to make, are driven by whole grains. This is one for you. We've been told to eat whole grains, whole grains, whole grains, whole grains, which is good, not a bad thing. But at the end of the day, whole grains may promote the growth of inflammatory bacteria. What the are we going to do now, So anyway, this is an interesting problem. Nobody's working on this, but we think we can get at something with the Hadza. Here's another group of bacteria that are associated with blunting inflammation. So when people start eating corn, okay, this particular small little group of people is a small, just, you know, this statistic doesn't mean anything, but it's interesting to us that they had a drop off in some interesting bacteria. Trep is another bacteria. It's associated with disease in the United States. The Hadza have dozens and dozens of species of Treponema. Treponema have been identified in Burkina Faso and the Mara Indians as well. We think it's associated with tubers or whatever. Treponema is really good at breaking down cellulose and xylem and stuff like that. So that's the difference between the wet and dry season. A totally different graph. I don't know why the hell I put it in there. Anyway, here's the Treponema after they started eating corn, which is we think in this particular case, Treponema is a good thing. In the Hadza population, it started to drop off. But this is the one that's scary. So you might think twice about, you know, I'm not a doctor, you know, Dr. Phil, Dr. Pepper, I'm none of that. Or not a medical doctor, actually, I should say. But this Prevotella is an interesting problem, and it's associated with whole grain consumption, and we see it in other studies as well. Turkey baster, again, I'm almost done. So what I did the first time I went out to Hadza land, I lived with the Hadza, I lived in their environment, but I went home every night and had my own food. I had pasta, and of course you guys can tell by now I drink. I had alcohol. I smoked weed, did all that kind of stuff. And so I lived in their environment. I took my stool sample every day. Okay, before I went to Africa, which I do anyway, which is weird. Maybe that's why I'm not married. Took my sample every day. And then when I'm in Africa, I collect my stool sample, freeze it in liquid nitrogen, and I see how being in their environment changes my bacteria. So I don't have slides for it, but basically just being in the environment, I started to accrue more diversity. I started uptaking different microbes, just being around them touching me, not drinking the water, not really eating their food. The second time I went out there, I went all in. Okay, I became a Hadza hunter-gatherer. I ate their food, I drank their water, you know, with baboon shit floating in it, the whole 9 yards, and then never get sick, by the way. And then that changed my bacteria as well. So what's left to do? I did a fecal transplant with a Hadza hunter-gatherer, and there's the turkey baster. So anyway, so before I knew I was going to do this, before I went out there, so this was the next step. In my experiment, I live with the Hadza. Okay, this is what happens. I eat their food, live with the Hadza, drink the water. This is what happens. Last thing to do is become Hadza. Okay, and so anybody that doesn't know what a fecal transplant, basically it's become an effective tool for fighting Clostridium difficile or C. diff infections. So if you're laid up in a hospital and you got your knee operated on, you got a bunch of antibiotics, and your Clostridium difficile said, "Woohoo! I just had scorched earth with antibiotics," and then they bloom up. They're a weedy species. They bloom up and give you some horrific diarrhea. You literally shit your brains out. And more antibiotics that they give you, it doesn't do anything for you. You waste away, and people die from it. It's a horrific, horrific disease. So what people started doing is they started taking fecal samples from a donor, sticking it up the yoo-hoo or running down the nozzle, the front end if you want, and then within 24 to 48 hours, miraculous recovery for a significant portion of the people that do it. So now it's become a pretty common tool for clearing C. diff. And the question that somebody asked earlier was, and basically what it is ecosystem restoration, all right? And again, if I tell you something you guys already know, I apologize, but I'll go over this really quickly. If you burn all the vegetation around us and you don't do anything to the outside but just let grow back what grows back, a lot of invasive and weedy species grow back. That's what happens when you take antibiotics. That's what happens when you go on a Western diet, okay? You're allowing weedy species and pervasive, non-desirable species to grow back. So if you burned the area around here and you went outside and managed what grew back, if you will, then that ecosystem restoration, you become basically a wildlife ecologist, you become a wildlife manager. So what you're doing is you're replacing the ecosystem, your sick ecosystem. So there's enemas and other things you do before you do the fecal transplant, depends on what your situation is. You're putting somebody else's healthy ecosystem, and then you're cured. Just like that. It's the most amazing thing in medicine in the last 100 years. Well, I'd say the last 50 years or 60 years since antibiotics. So now what they're saying, well, if I've got IBS or I got Crohn's disease or I'm too big or I'm too little, I'm too thin, maybe I could do maybe I want your bacteria in my body and make me thinner or get rid of my IBS or get rid of my autism. There's an autism study being done right now where they're doing fecal transplant with autistic kids. There you go. Okay? So there's a lot of it being made of it. Most of it is bullshit. I can say shit in here. But it's interesting. It gives us a methodology or a way to think about changing or shaping or modulating your gut microbiota. So I talked to tons of people about this before I did it. Not a single researcher I contacted. And then this project has a lot of researchers on it too. Every one of them said, don't do it. Don't do it. And so Rob Knight, the New Zealand guy that looks like a woman shopping for real estate in Florida, he said, don't do it, but if you do, it'll be really cool. It'll be really cool. And I want to see the data. So I got hold of Marty Blazer at New York University. Marty was out in the field with us about a month and a half ago. He's the chair of the medical department at NYU. He just got a Lifetime Achievement Award from the American Infectious Disease Association, too. Nice guy, really smart. Anyway, I told Marty I want to do this. He said, hell no, don't do this. What do you need me to bring? And I said, well, I need a turkey baster. So there's a picture of him in Walmart. I can't find the damn picture. And he's got this turkey baster. It's like this big. He brought me the biggest turkey baster they had. So anyway, before I forget, I wanted to mention this. There's a group of bacteria called Oxalobacter. Okay, it's a particular group of bacteria that pretty much everybody in this room doesn't have. All the Hadza have it. 75% of them have it. Oxalobacter has now been connected to kidney stones. So you have oxalates, which are in certain foods and so on and so forth. And if you get oxalates in your gut, the oxalobacter eat it and blah, blah, blah. If there's no oxalobacter bacteria, the oxalates end up in your bloodstream. They end up in your kidneys. You have kidney stones. Marty, again, should win a Nobel Prize for this if he figures it out. So there's 2 guys on the project that should win Nobel Prizes. So the Hadza have oxalobacter. And lo and behold, I accrued oxalobacter just by being in their environment. So if you have oxalobacter, probably not going to get kidney stones. Anyway, so the Hadza are interesting. He has oxalobacter, he does, but one of them doesn't, so 75%. So anyway, one evening here about a month ago, I sat underneath the baobab tree, took my donor with me, already knew what bacteria he had in him, didn't know on the parasites yet. We didn't have the data back on the parasites yet. We took him to the hospital. We had him tested for Hep A, Hep B, Hep C. Taryn administered rapid field tests for HIV. She tested him for HIV, and I said, test him again. She tested him again. I said, test him again. She tested him again. Tested him the next day. And then I tested me. I was like, oh, shit. So I tested me. Everybody's fine. But I didn't know about the parasites. And basically, the risk I ran was getting a virus that I didn't know was there or that my immune system would freak out. And at the end of the day, everybody said, why? Why? You're not sick. You don't have C. diff. Why? Why would you do this? Why would you do a fecal transplant? I'd done the diets, okay? I'd done that. And so it was the next logical thing to do. So the question is this. I did the fecal transplant. I laid underneath the baobab tree. He gave me a Tupperware bowl full of his dew. I had it in my body within 120 seconds, okay? You got to get it quick because of the anaerobes and so on and so forth. So I'd put it in, shook it up, got it in the turkey baster. I lubed the turkey baster with some organic lube. I went... Too much information. We're all friends, though. That far. Juiced it. Laid on my back, put my butt on a rock, and pedaled an imaginary bicycle for 30 minutes. That was at 7 o'clock at night. I watched the sunset. A tear dropped from my eye. Watched the sunset. Watched the sunset over Lake Eyasi. Watched the Southern Cross rise in the sky before me. Was emotional. It really was. No kidding. A little bit emotional. So I did it. I didn't go to the bathroom until 11 o'clock the next morning. But the minute I finished the fecal transplant, of course, he gave me the Tupperware bowl. He handed me the Tupperware bowl, and he walked about this far, and he was like, he's like this. He's like, what are you going to do? It's like, I don't speak Kadzabi or Swahili. I don't speak enough Swahili to understand, but he just went back to camp. He's like, well, whatever. And so anyway, so I did the fecal transplant, immediately went back to camp. I'd been on the Hadza diet for a couple of days. I've been eating baobab and stuff. I wasn't smoking any weed and stuff. You guys can tell I drink and smoke weed. I hope you're not cops. And so I immediately went back to camp and started eating Western food. So what I wanted to do was, I have the samples now. They're at NYU, or they're going to be analyzed pretty soon. So I had samples before. I had your samples before. Taryn drew blood. We had blood as well. So what I wanted to see was, could I replace, effectively, if it took, okay, the Hadza have the most diverse gut microbiota in the world. If my body accepted it, I have the most diverse gut ecosystem of any white person in the world. Okay, any white person in the world. So anyway, so what I wanted to find out is, could I immediately go on the Western diet? Could I come back to the United States? Could I eat cheese from Shelburne Farms? Could I drink dry margaritas or whatever those things, martinis were last night? Could I eat chips? Could I destroy that microbiome? Could I destroy it? Okay, so what I've been doing ever since is I've been trying to destroy it. So if I acquire, and I'm on a tear, Anyway, let's say I picked up 2,000 OTUs from this guy, okay, and I only had 1,000, so we don't know what took, maybe 60%, 80%, who knows what took. So I immediately took stool samples in the field afterwards. So we know what it looked like before, during, after, blood, urine, everything. Everybody's like, how do you feel? You feel great. Taryn told me I farted a lot less. So I got back. So now I go back in November. So I keep taking stool samples. I sent one in the other day. I go back in November. When I go back in November, I'll know what the results were. I'll take another fecal transplant, okay? And then I'm going to see, but actually before I take the second fecal transplant, I'm going to see if I go back on the Hadza diet and live in their environment. If I could, let's say he had 2,000 species, can I get all of them to come back up again? Okay? Or did I undergo a mass extinction? So what I just did, if I don't screw this up, I basically recreated, you got to squint a little bit to believe this, like everything else I'm talking about. But anyway, did I just recreate 10,000 years of human history? You know, did I just knock the crap out of my microbiota with my Western diet and lifestyle, riding around in a rental car, inside a room with windows, drinking chlorinated water, haven't taken antibiotics or anything like that, doing everything I can to pick it up, okay? But does it bloom back up? If it doesn't bloom back up, fascinating. If it blooms back up, fascinating. Doesn't matter. The next time when I do it, in November, I'm going to do another fecal transplant, but then I'm going to stay on the Hadza diet for weeks and weeks and weeks and see if I can maintain it. I effectively replaced 99.999% of all the genes in my body with his. So I'm more Hadza than I am Texan. So anyway, say again? Did you end up feeling better? I didn't feel bad to start with. You know, but when people say, why would you do it? Why are you sick? I'm saying, you know, you're not sick. I said, the fuck, I'm eat a Western diet. Of course I'm sick. I have less diversity than he does. I'm sick just because I don't have C. diff or IBS, which is the measurement of being sick. We're all sick. We're all sick. So for me, it was worth the risk, but it also goes to the core of why I do all this, is to try to understand why my daughter has an autoimmune disease. So I need to understand extinction. We now know that everybody in this room has lost a third to a half of all the microbial diversity that your ancestors used to possess. A third to a half. At least. So as simple as it sounds, so you can take up all the disciplines in medicine right now. You can take all the fancy names and all the subdisciplines and every this you should be taking and every omega that you should be taking and this you should be doing. At the end of the day, it could be something as simple as reintroducing microbial diversity back into our life. But we've got to ground it in solid science. We're not arguing that we all have to eat a paleo diet. These paleo hunters were vegetarians for weeks on end. So we can argue all day around about how much of this or how much of that or sugar and all that kind of, you know, it may just be something as simple as reconnecting, okay? Maybe reconnecting. Yes, ma'am?
Yeah, I was wondering what was happening to their blood sugar during the 6 months that they were living on honey.
We didn't collect blood during that. We don't know. Yeah. I'm sure it shot through the roof. Yeah.
But no diabetes in that population?
Nope. Not at all. None. So anyway, I'm almost done. This is it. Time is running out. That's a Hadza hunter-gatherer. That's his corn harvest. His name is Ezekiel, okay? So what's happening to the Hadza now is we know that 10 to 15% of the Hadza have taken antibiotics now. Some camps it's a little higher, some camps a little lower. In the next 5 to 10 years, 100% of the Hadza will take antibiotics. They have access to it. It's over. It's over. We cannot study this population after that happens, okay? In terms of looking for it, so what we're doing is trying to launch projects in other countries too, but this is the ideal population in the world. So that's why I spent months there last year. I was there in January and February. Tara and I were there for 4 months over the summer. I'm going back in November. So we're raising money to buy beans and rice and diesel, doing everything we can to collect more samples before it's gone, okay? So they're losing the hunter-gatherer life way. Now we're studying mothers. We're collecting breast milk. We want to be able to study babies from birth. The minute that baby's born, the minute it's born, we want to sample it every day for 2 years. Every day. We can learn more from one hunter-gatherer baby, or 5, born in the dirt, in a hut, than we can learn from 1,000 in the United States that we spend millions of dollars studying. It is the most, not just because I'm doing it, I believe it is the most important microbiome project on the planet. And it should receive priority at some level, somewhere. So now that we've put together a who's who of researchers on the project, we've raised a little bit of money from philanthropists. Now we're going after NIH for some money. Those boneheads will take 2 years to maybe finally wake up and give us. But meanwhile, he's growing corn. Meanwhile, he's charging his cell phone. Meanwhile, he's trading honey for cooking oil. Or he's trading honey for booze. or he's training honey for more marijuana. Not a bad thing. This blood, that man's arrows, and those tubers and everything are what's important. So we're going to be doing this for the next couple years. Anybody wants to donate $5 to the project, go to humanfoodproject.com. $5 will buy 1 liter of diesel in Tanzania. And that's it. That's all I got. Anybody got any questions? Yes, ma'am, in the back.
I want to know if you're going to introduce antibiotics to your house or not.
No, ethically we can't do that.
No?
Well, we're not supposed to. But here's the problem: is when we're in a camp, like Tara and I was there at camp, people die. People are dying. People have infections. So a lot of people administer Cipro and things like that, but we don't do it in a controlled way where we're doing an intervention. It's against IRB rules. But you'll find other researchers who work with the Hadza, anthropology stuff, will administer medications.
But your house,
Oh, mine. I might. I'm thinking about going to Mexico and getting some teeth work done. And I'm thinking about I'll have to take antibiotics. But I want to wait until I get back in November because I want to do it again. But that would be a great test to administer the antibiotics. Yes, sir.
Cryogenics, you're freezing this stuff.
Yeah, -80 and liquid nitrogen.
What dies off in cryogenics?
The problem with the liquid nitrogen, there's been tons of control work done. Basically what happens is it's freeze-thaw that's the problem, okay? So there's very little degradation at -80, but if you let it thaw and then freeze it again, you might as well throw it out the window.
So can we collect this stuff and if we put it under ice, so to speak, to save?
That's actually a good question. One of the things that I wanted to do when we started this project was we knew these guys were going to be disappearing, so we collect these little cryo tubes, okay, like these little bitty tubes, like that big pencil you had when you were a kid in school that was that big around. Cryo tube, fill it full of poo. And for the 16S work, you literally just need a speck of shit, literally a speck. So we have all this biomass left over. So our argument was that we would store this biomass. 20 years from now, there's going to be a hell of a lot smarter people, better analytics, better equipment, better questions. So we want to be able to have the ability to go back to these samples at some point in the future. Okay, so that's what we're doing. We're biobanking these samples, a lot of them. Yes, sir. Yeah, they do. So when they eat the wild honey, you eat the cones. That's a good question. They eat the larvae too. So it's the most energy-dense food on Earth, but when you add the larvae to it, it goes up another tick. So yeah, they eat the larvae. So it's high fat, high glucose, fructose, and it tastes fantastic. It's unbelievable. And there's lots of, everybody knows this, antimicrobial properties to honey with regards to skin infections. So we're actually going to, just so you guys know, at Stanford University, we're going to take Hadza poo and put it in germ-free mice and then do things with the mice, not going to play with them. We're going to do stuff with the mice. We're going to study them. So we're going to look at Hadza gut in the dry season versus wet season. Then we're going to challenge the gut with honey, challenge the gut with different fiber and stuff. So we're going to be humanizing germ-free mice in Stanford with Hadza poo.
You said it tastes good. Is there a particular honey, like particular tree that it tastes the best?
Taryn could probably answer this better than me, but it tastes different. It depends on the flower, which is the pollen. You have the big baobab trees, you have the stinging bees, and then they have a little bee, it's called quinoa. It looks like a gnat. It produces this honey. It comes like in little bands, like a cigar. It's the best tasting honey you've ever had in your entire life. But they eat the pollen. They eat everything. The bee is called quinoa. It looks like a gnat. It drives you crazy. But the honey's really good. So, yes, ma'am. Basically sterile water. I mean, there's 1,000 different ways to do it. I just put it in some sterile water, some water that I'd boiled, shook it up, and got it into solution. And
Are you then able to retain all that?
yeah, so I puckered up pretty good and I put it in. I squirted it in, pulled it out, and stayed puckered. And it stayed till 11 o'clock the next morning when I had my first bowel movement. And of course, I sampled that bowel movement. So in theory, I mean, it was in there. So whether or not it took or how much it took, I don't know. I will know in about 2 or 3 weeks.
One other question, if you don't mind.
Yes, ma'am.
You said that there are certain bacteria that are associated with lean people and certain bacteria that are associated with
It's a tough question. I'm probably the wrong person to ask, but basically at a 50,000-foot view is more obese people had firmicutes than they did Bacteroides, which is like Ford and Chevrolet, all right? So that was found out in 2005. Doesn't really work that way anymore. I have 80% firmicutes, okay? The Hadza are 80% firmicutes, and they all look like Ben. And so, yeah, so it's a little bit more complicated than that. There's, you know, maybe it sounds like you know a little bit about this. You know, there's association with gram-negative bacteria. There's a thing called endotoxemia or low-grade endotoxemia, which Patrice Canny and the guys in Europe figured out. If you had a certain group of bacteria, gram-negative bacteria, that produced basically lipopolysaccharides, I'm oversimplifying it, basically bacterial skeletons, I'll just call it, that are leaking into the serum that are causing low-grade inflammation. So people that have groups of this particular gram-negative bacteria seem to have more things floating around in their blood than they should have, and a little bit more low-grade inflammation. So it's been done. It's being tested in clinical trials and stuff now. But so seems to be some groups of bacteria, but the simple distinction of firmicutes to Bacteroides doesn't really work anymore. But there is a group in there that's causing some low-grade inflammation. That's a leaky gut. You know, everybody talks about the GAPS diet and so on and so forth. But if you can figure out how not to get LPS in your blood or LPB or LPD or whatever in your blood and shut down. And that's one of the things I can do is I can monitor that in my blood after the fecal transplant as well, and I can monitor it in my urine too. So to answer your question, I really didn't answer it, but yes, there appears to be some guys out there you may want a little less of, but nobody's going to go out and say it's this group or that group for a long time, but I will as soon as I figure it out. David. Say that again? Resistant starches? Yeah, RS got really big lately. You know, there's a lot of people talking about, and there's RS 1, 2, 3, 4. I don't pay a lot of attention to resistant starches. I know that RS2, resistant, like potatoes, you know, have resistant starch in it. They will feed your bacteria, but whether or not they modulate it in a particular direction that's useful, just because they produce short-chain fatty acids doesn't necessarily mean you're growing the best bacteria. But anything that grows short-chain fatty acids and makes your colon more acidic, I think is a good thing. But it's an interesting area of research, and I think the paleo community's kind of glommed onto it a little bit, but it's not really well founded, at least from the perspective of the gut. There's a couple of guys working on it, but so the answer is I just don't know. But resistant starch in the Hadza diet would come from tubers. You know, they dig a lot of subsurface tubers there, but it's not like a potato. It tastes like a potato. A root called equa that they eat. It's my good Swahili, equa. And, but, you know, it's got a little bit of resistant starch in it. But, you know, again, there's not a lot of resistant starch, I think, overall in their diet. They eat a lot of fat, a lot of protein, and a lot of sugar, and a ton of fiber. One last comment on fiber. If you look at the U.S. Department of Agriculture's dietary recommendations for Americans, 2015 is about to come out. It's 13 experts right now at this very moment trying to figure out what recommendations to make to HHI and USDA. And of those 13 experts, not one of them is a microbiologist, not one of them. And we now have this huge connection between diet and microbiota, but the microbiologists don't want to come out and say anything. They don't want to get burned. Like in 1995 and to 2000, when all the guys were trying to raise money to do genomic work, they promised the government that by now we'd cured every disease. If you just give us another $1 billion for research, we'll get to the genetic basis of every disease. Well, it didn't really pan out. So microbiologists literally hold in their hands, and this is not an exaggeration, an overstatement in any way, shape, or form, microbiologists and microbial ecologists and bioinformatics people and people who worry about bugs on a daily basis literally hold the health of the planet in their hand. When I say health of the planet, microbes are tied to CO2, microbes are tied to water temperature, they're tied to nutrient turnover, they're tied to everything, and we're just one cog in that wheel. But within the next 10 years, next time you go to the doctor, they're not going to draw blood, they're not going to take urine, they're going to take a poo sample, and they're going to put it in a machine that looks like an ATM that had a love child with a copy machine. And that's what it'll look like. They're going to put it in, and 2 minutes later, it's going to spit out your microbial profile, and they'll tell you exactly what your blood pressure is, they'll tell you exactly what your cholesterol level is, and they'll tell you exactly which way you need to nudge your microbes. The fear I have is this, and it's a real simple one, is we've all kind of now started to take our health back from doctors. You know, now we have alpha nutrition females in the family. We let food be our doctor now, so to speak, or be thy medicine again. We're taking it back from pharmaceutical companies. I saw a horrifying word about 2 years ago in Nature, or it might have been Science, and it was a guy named Jerry Nicholson. He's at King's College in London. He said, there will be a day when we drug the microbiome back into compliance. And here we've been handed this wonderful gift that you actually have within your power by your actions, your diet, and where you go and who you hang out with and whether or not you kiss a dog at night, and I don't mean your husband, is that you have within your power to nudge your microbes in the direction you want. You do not need to go down to GNC and buy that. You do not need to go to your doctor to get a prescription unless you have something like C. diff or something like that. And so, so now we've been empowered with this ability to nudge it. We just need to know what direction to nudge it, okay? So once we understand that, we don't need to drug it into compliance. And once we understand it's about ecosystem restoration, we understand the basics principles of Ecology 101, then you just scale that up. Now the global argument about ecology starts to make sense to you. I think the microbiome has the ability to turn the entire planet into a bunch of ecologists. And as soon as you can do that, then we can wrap our little skinny heads around the bigger problems. It starts with your gut, and then Greenland starts to make sense. Carbon emissions start to make sense. It's about ecosystem. It's about steady states. It's about equilibrium. So we need to teach this shit in the classroom. We need to talk about it more. And I think the gut is going to lead us all down that path, willingly or unwillingly, as we move. But we can't drug it into compliance, which is, I did it to some extent. I took my own action. I took things into my own hands, not because I was sick, but, now I possess, if anybody wants a piece, the most diverse ecosystem in the world. I could raise money like that. 1995, I'm in the West Room.